This is a production of Cornell University.
Hello everyone, my name is Michael Gore. I'm coming to you from this section of plant breeding and genetics. So the question of the day, well perhaps this isn't a question of the day for you, but it's a question of the day for me that I will pose to you, is that how did Mike Gore get into high-throughput phenotyping? Let me go back to my days of being a graduate student in Edward Buckler's lab. We had this population of 5,000 recombinant inbred lines called the Mays-NAM population. And it took an army of people to plant and an army of people to phenotype. So after all those countless days of having to do plant height, ear height, or flowering time out in the field, I began thinking of ways to do high-throughput phenotyping. One of the ways that we had thought about doing high-throughput phenotyping was perhaps having a plane fly over the field, which we had. The other way was having a large helium balloon tied to a four-wheeler and pulled through the field, and a nice expensive camera attached as well. We began doing that and driving back and forth through the field, but sometimes the wind would pick up and there would be some tension on that line. It was a little scary, but we were all pretty sure that that line was strong until one day it snapped. And I remember our field manager, Nick, calling the police and asking them if they would shoot the balloon out of the sky. I'm so thankful for humanity that they did not do that. And for all we know, that camera is in Canada somewhere and it's being used. But thankfully, I didn't allow my high-throughput phenotyping dreams to float away that fateful day. So in 2009, I accepted a position to work in the USDA in Maricopa, Arizona, which was a great decision on my part. The environment was very harsh for human and for growing plant, but I kind of viewed it as a test track for plants. And it's a great environment for testing for heat and drought stress. And I was hired to work on cotton, so coming from the maize community and at that time working on the first generation haplotype map of maize, realizing I was going to have to dedicate my life to working out in the field, maybe the only people I would make proud would probably be Mark Sorrells, because I would actually go out to the field. What I didn't realize at the time when I had my interview there in January is it gets really hot. And when my family and I moved back out in June, there were so many four-letter words dropped that day. But you know what? We adapt just like the plants. But some of the challenges, such as when you think about cotton, trying to sustainably produce it, when in the face of global climate change, the patterns of weather are highly variable right now, and our freshwater resources, especially when I was living in Arizona, when we were drawing down the Colorado River, when you actually see it firsthand, it is quite traumatic. And here, when we're looking at the cotton belt right here of the US, and this was taken in 2011 here, this is where there was a co-occurrence of a severe drought. And just to have an idea of like the economic impact of what a drought can have, this caused $2 billion of economic loss in cotton production. What are some of the consequences of that? We see it on the news, but really it's impacting human beings. And so when I was hired, this is kind of the charge that I felt burdened with, that I felt I had to carry, is how are we going to turn that engine of evolution in plant populations fast enough to generate these resilient cultivars to help provide fiber to the world, but also provide a source of financial income to the farmers, such as this one in Garfield County, Texas. And in 2009, we began thinking a lot about genomic selection. And of course, we could also think of recombination and CRISPR-Cas9 tying into a genomic framework. And the whole idea is how many more generations can you turn per year? And so ideally this would result in faster development of more resilient crops, and in this case, cotton. So of course we have our training population, and we do genotyping and phenotyping on that training population to train our GS model. But phenotyping is still very important because when you think about the prediction accuracies, highly heritable phenotypes and high quality phenotypes is what's driving a lot of that GS model. And so there's obviously a lot of improvements that can be made to these GS models where you're training on a genotypic and phenotypic data set, and then you're trying to predict the phenotype of a related population that has been only genotyped. And phenotype is what I decided to focus on at that time, back in 2009. But coming into it, we all knew that the classic phenotyping, as I talked about earlier, plant breeders know how to do very well. Plant breeders are very industrious in a sense. They'll just hire an army, and that's how they get the job done. But when you're working in an environment such as Arizona, which has sometimes temperatures over 100 degrees Fahrenheit, you need to work a lot more intelligently or you won't last very long in the field. So there's that time-intensive component to it. Often, if you're doing plant physiological traits, the sample sizes are fairly small, and you can only do perhaps like a few time points as well. And often, from a plant breeding eye, there's obviously trait bias in a sense. You tend to only phenotype what you can visually score, or perhaps you're just driving a combine at the end of the season. And when it comes to hiring a large field crew, that is also very expensive. So this is my colleague here, Mike Salvucci, and he introduced me to the Lycor 6400. I was all enamored with it at first until I realized how much of a pain it was having to work in that intense heat, and to realize you would sit maybe for a half an hour in front of a couple plants and you wouldn't really get that much phenotypic data. But some of the phenotypic data that we were able to get would be like transpiration rate and canopy temperature. And we also had a FLIR camera that we could shoot in front of these. And here, this is pictured in Pima cotton. If you do not know what Pima cotton is, it has nice long fiber, and that's like that high thread count sheets that you have. But at that time, we were really thinking about how do we do this a lot faster? It isn't that we're lazy, we just want to have very large populations, and we want to be able to really cover all the different developmental time points of a plant as they are growing over the growing season. And we want to be as objective as we can possibly be, and we want to do this at low cost. So there was an agricultural engineer that I began talking to in 2009, Pedro Andrade Sanchez, and in a sense we found this tractor in the junkyard, and it needed a new engine and new tires and everything. So here I'm showing, I think at this time, version 2. Version 1 tractor, high clearance tractor, we just pulled out of a junkyard and put in a new motor, put some new tires on, put this toolbar on, and we're set to go. So the idea of sensor fusion is, in a sense, combining all these different data streams to be able to use them and get as much information out of in terms of trying to understand what is the crop doing over these various time points of the growing season, and if you're imposing, in this case, heat and drought stress. And some of the phenotypes that we were focusing on first was a vegetation index, and that's focused up on here, and these are called crop circles. We had IRTs on the tractor as well, and they are pointing down over the crop right here, and that's to do phenotyping of canopy temperature. And here we have the ultrasonic transducers, that's pointed right here, and that's what we were able to do, canopy height. And we had RTK GPS on the tractor, so in a sense we're able to resolve and deconvolute all these phenotypic data points down to individual plants in a plot, and we're able to do that entirely and repeatedly over the growing season. So the first thing that you do kind of as a plant breeder is think about, okay, we're able to collect all this perhaps gigabytes and terabytes of phenotypic data, but is it of value? Is it heritable? Is it repeatable? So one of the first things that I basically want to do is here we were testing this on 25 Pima cultivars, and here we have on the y-axis canopy temperature, and on the x-axis leaf relative water content. And what this is basically showing is this canopy temperature is tracking with changes in the soil, plant, water relationship, whereas the dry plots have a higher canopy temperature because the transpiration rate is reduced of those plants because they're trying to conserve water. So the transpiration rate is shut down, and in turn the canopy temperature is increasing. And here in the blue, these would be, and you can kind of view them as control plots. So these are the well-watered wet plots, and the transpiration rate is high because these plants aren't shutting down, and they're able to continue to cool the crop canopy. And we had heard from Todd about this vegetation index called NDVI, which ties in red and near infrared. And what this is really typically doing is, in a sense, quantifying biomass, but it's also tying in leaf area index, chlorophyll content, and coverage of the actual plant canopy itself. But what we saw here over time of these 25 Pima cultivars is the plots that are dry, they begin to decrease their NDVI value over time. And what this response was actually capturing is the degree to which the plants wilt. Because here you see a Pima cotton cultivar that's under intense drought, and you can see the leaves wilt. So this is a vegetation index that is, in a sense, capturing greenness. So as the leaves droop, there's more soil exposed up through the canopy, and also as the leaves wilt, they're changing their conformation as well, and that's changing the pattern of leaf reflection as well, and that's why those values are decreasing. So this was like kind of a fun way to see if you can plot this over time within the actual day. Is capturing biomass, but in our eyes now, we're able to use it to see the degree to which the plants actually wilt. So we were able to also see that this system was repeatable over the growing season, assuming that the canopy was closed and that the drip irrigation was not clogged. Because there are challenges when you're growing on drip irrigation and it isn't cleared out right. You need to pump some sulfuric acid through there and clean it up just right, and things begin flowing very well. But as we saw is, when we were doing this high throughput phenotyping with the tractor, we could easily see parts of the field from the response of the plants where the drip tape was actually clogged, because those were the plants that were heating up very fast. And then you would just pump in the sulfuric acid and clear it out, and it cleaned it up just fine. And so here we have the same high clearance tractor system, and here I'm calling field-based high throughput plant phenotyping system. And it's QTL mapping of different traits over time. So if you haven't thought about QTL in this sense, you can just think of a QTL as a quantitative trait locus, but in a sense it's just a chromosomal region controlling phenotypic variance in that part of the genome. So in a sense, we have this recombinant inbred line population of close to 100 recombinant inbred lines, and these recombinant inbred lines are a mosaic of the two parents. So in a sense, you shuffle up the genome, and then you're using the patterns of recombination to track these regions of the genome with the phenotypic variation. And if those particular regions of the genome are showing up, that's what we're referring to as a QTL. So here we also have our field-based high throughput phenotyping system. We did these various agronomic traits such as lint yield, boll size, and fiber quality. And thanks to Tim Setter at the time, we were also able to phenotype abscisic acid concentration as a physiological leaf trait. And as I was saying, this was all grown in drip irrigation, and we had soil neutron probes in the field, which if you've ever put these in, you can feel our pain, because we would have to do these readings every week before and after turning on the drip irrigation. And the great thing about having a drip irrigation, you can have a set time. So in a sense, the plants are almost trained very well when you're turning on and off the drip irrigation. And this is a shot of the actual field right here in Maricopa, Arizona. So here, what this is basically recapitulating a canopy temperature heat map of the field. And each of these little circles or pixels right here are an actual phenotypic data point that was taken by the high clearance tractor. And so if you recall, we had these 4 IRTs on the tractor, so we're able to do phenotyping of 4 rows at a time. And the cooler the canopy is, the bluer it is. And you see these little areas right here, those are alleys in the field here. And so this is a control treatment right here. So here would be the dry drought treatment right here. And here you can see the canopy temperature is heating up. But you can see this part of the field right here is actually cooler. And when we ran an EM38 and checked out the conductivity of the soil, what we had found out, this part of the field had a higher clay content. And so if you do any drought testing in the field, the thing you should always understand is there's more variation for the soil than there are actual traits that you're phenotyping in your plant populations. So you have to control for this type of field variation in your models, or you'll just end up being a breeder breeding for soil type. And you wouldn't be the first one that has. So the great thing here is that canopy temperature turned out to be highly heritable in this recombinant inbred line population, and in this case shows transgressive segregation. And what that transgressive variation actually means is you have progenies that exceeds the phenotypic values of the actual parents. And these are the 2 parents here, NM2416 and TM1. And these are the control plots here, wet. And these are the dry plots here in red. And you can see the control plots here, wet had a heritability broad sense of 0.83, and this is heritability of 0.9. These heritability values generally range between 0 and 1, and they happen to be quite high here because one thing to keep in mind, we were doing phenotyping at 3 time points within a single day. So it's really taking in all of that variance within the actual day, and that's helping to increase the heritability of the phenotype itself. And since we have the tractor, we can do phenotyping over the entire growing season here. So if you haven't thought about cotton growing, so here we go from seed to perhaps 8th node, 4 to 5 weeks after first bloom, and then you have to add a defoliant to force all of the leaves to fall off, because cotton is a perennial tree that's treated as an annual crop, okay? And the great thing is, here's when the trials are planted. We have equal irrigation applied to both the wet and dry treatment, and these are the phenotypes that we had targeted at 4 different time points, so canopy temperature, canopy NDVI, which, just to remind you, is a vegetation index, and then canopy height. And then we began opposing the differential irrigation at flowering because we didn't want confounding, because that's why we didn't do it early on prior to flowering. Because also, as a few of you probably know, often when people are breeding for drought tolerance, it's actually drought escape, and the QTL tend to be just genes for flowering time, and that's one thing that we wanted to avoid. And then at the end of season, we're driving like a cotton picker through the field and harvesting. And so here, what we have is canopy temperature over the whole growing season, and then we were able to break up these basically days of the year into the different developmental growth phases of cotton. So here we have flowering at peak bloom, bulb development, and fiber development and elongation. And this is at 7 in the morning, 10 a.m., 1 p.m., and 3 p.m. And the red here is the dry treatment, and the blue is the wet treatment here. And what you kind of see on this axis here, this is the canopy temperature here. And as you increase the temperature in terms of during the day, the phenotypic variance is also increasing as well as the plants are becoming more hotter. And so when you think about this, we were able to now break up the plant into these different developmental phases. And what we found is the canopy temperature phenotypes themselves were more highly correlated within a plant growth stage. So what that's telling you is there's somewhat maybe of a coordination of genes themselves within an actual plant growth stage. So then this gave us the idea, okay, since we're seeing these patterns, what that may suggest that there is maybe certain QTLs in terms of their expression patterns turning on and off in the genome over the growth phase of the cotton recombinant inbred line population, and some of that is perturbed by heat and drought stress. And in fact, that is actually what we found. So here we have, what we're revealing here is this distinct temporal patterns of the QTL effects that's controlling canopy temperature variation in this recombinant inbred line population. So this is the QTLs that we detected under the control wet treatment here. These are the QTL that we controlled under the dry treatment here. And these are the effects of the QTL that decrease canopy temperature here. They're in blue. And in the red are QTL effects that are increasing canopy temperature. But what you can see here is, depending on the age of the actual plant, that's when QTL are actually being triggered and turned on. And the intensity, if you will, the size of the effect of the QTL is also partly being driven by the drought treatment. And the great thing about a genome is you can find any possible candidate gene that you want. So here in this region of the cotton genome where we had a QTL mapped, we found a gene for an ABA response factor. And one of the things that I found was pretty exciting at the time is that the QTL for variability in cotton leaf ABA content also mapped to that same region of the genome. And why that seems to jive is, in a sense, when you have ABA, it's contributing to what? The model size and how it's going open and close, right? So it's quite possible that this QTL is, in fact, contributing to variation in transpiration rate in this part of the genome, and that would obviously then drive changes in canopy temperature. And Dr. Duke Pauly contributed to this paper. I believe he is here. You can ask him all about it. And so then we also want to think about in terms of breeding. And when here on the x-axis here we have lint yield and temperature on the y-axis, what we have found is, that the increase in leaf transpiration rate under drought stress actually contributes to higher lint yield in a heavily irrigated hot air environment. So these plants over here are the ones that take a risk under drought. In a sense, they're not shutting down and they're continuing to have a high transpiration rate. And by having that, they can continue to fix carbon and, in a sense, have higher yield. These plants over here are the ones that decide to shut down, and they're going to fix less carbon now, right? And so they will yield less. So in a heavily irrigated environment such as out west in Arizona, you would perhaps want a genotype such as this one right here. But if you're in a dry land environment that's only rain-fed, perhaps you would want a genotype that's more conservative over in this region here, and that will perhaps be calibrated to use water only perhaps at like fill of the grain or at flowering. And what we also found is at all these various time points that we did phenotyping across the field season, the most informative was at flowering. Because the reason why is, after you have all this heat, what's going to suffer? In this case, it's going to be the flowers themselves. So if there's high heat, the pollen isn't going to be viable, and if it isn't going to be viable, there isn't going to be seed set, and there isn't going to be any fiber. And so here is the fun part with Lidar. We're putting this on our tractor, and now we're trying to recreate the 3D shape of defoliated cotton plants in the field. So this is our entire field right here. And this is one, say, like 28-foot plot of defoliated cotton plants. And what you can see here is all of these cloud points, and Todd was touching on cloud points in his talk. And in a sense, these are recapitulating the actual cotton bolls on the plants. And we can actually take out like a single plant and do a Euclidean clustering approach, and in a sense try to get closer to yield. And we're just counting the cotton bolls on the plant. So I think there's a lot of future in that. So now let's turn to deep learning, which we all love, and artificial intelligence. So since coming to Cornell, I've been working with some plant pathologists, which I found are exciting people to work with. And, oh, hey Chris, how you doing? So one of the questions we had, here we have a phenotype that's caused by this pathogen, and this disease is northern corn leaf blight. Say you hire maybe 4 or 5 undergrads, you show them one leaf, and you force them out to go to the field, and they have to now do phenotyping for NCLB. Well, you just showed them this one, which has NCLB, but they see all these other examples here. Wow, wouldn't it be so much easier just to train a robot? Well, at least that's what I thought. But obviously, it's important to invest in our youth of tomorrow. So this idea is training these convolutional neural networks. So this is what you refer to as, in the old days, deep learning and now AI. So the idea is to combine this with drones to detect these NLB lesions on maize leaves in the field, and to turn drones to just, for just being something fun you see flying around or trying to tear out of my kids' hands when we're going to target, is that you're actually turning them into a coworker and a colleague now. So the idea is you have your drones flying over top of plots of maize, and you have your RGB 3DR iris cannon here, and then here is a disease lesion. But obviously you don't want to take, as was shown earlier, perhaps like 80,000 images and try to pick these out by eye. You want to train these CNNs and to detect NCLB in maize. The challenge is we haven't evolved to the point where the AIs can train themselves, but I'm sure that's coming in the future. So here you can just kind of view it as the CNN here is converting pixels into words. And the awesome thing, it doesn't need to be linear. So you can use curves and shapes and everything like that. But this is where presence absence and a human being, trained plant pathologists, have to classify this is a disease lesion or it is not. And I've been told by the colleagues on this project, after they would do maybe several hundred of these per day, when they would close their eyes, they would see these disease lesions on the back of their eyelids when they would go to bed at night. So I do feel kind of horrible for that, but it's all in the name of science. So here we have, this was taken from, if you will, a person as they walk through the field with a handheld camera. So you can kind of view this from the point of view of like a ground vehicle. And we also have all the ones from the drones that we're training on right now. And this case, we trained on close to 2,000 photos that have been classified. So here, we have our AI, and these are all our disease lesions here. And our AI, this is kind of viewed as a heat map, and if it's white, that means it has a probability, of, in a sense, that is a disease lesion. If it's black, there is none where it's black. And so that's when they are present, and if they aren't present, then you should have an AI trained very well so that you shouldn't detect any lesions when there are none present. And in this case here, we do not find anything at all. And after training on these 2,000s for this set that we have, we found maybe we're only wrong maybe 2 to 3% of the time. So I think we're doing pretty good. Perhaps when we train on 10,000 more, we can maybe increase that even more. So this is like a very fun, active area of research in my lab. And on our bread project, it's kind of the same concept, but now changing to our handheld phones. And this is a colleague of mine where he, in a sense, has developed an AI to detect the disease that's infecting like a cassava plant. And in this case, a farmer or a breeder can take a picture of a disease cassava plant, and then the AI runs on the phone, or actually it can be sent to the cloud where it's processed there as well, and then with a certain probability it'll tell you what cassava disease it actually has. And in the future, for our Terra project that we have, we've kind of taken the approach for trying to develop low-cost autonomous field robots that can go through the field. And here with our colleagues at University of UIUC, the idea here is, so you can do canopy reflectance, canopy temperature, and canopy geometry, and it's on-board processing as this can transect like the plots in the field, and it's recording other climatic factors such as like soil temperature, air temperature as well. So our first like version one is it'll cost about $200K, but we're very hopeful that we can get the price down to like $20K, and then we can kind of think of a world where we have these in everyone's hand. And then, so we're testing this on biomass sorghum, and in my group we're creating a hapmap for a few hundred lines, and the idea is we can then develop like a time series for like G by E at all these variants in the hapmap across the genome, and see like genes that are being turned on and off, as I was showing with like cotton, but really beginning to factor in that G by E component to try to figure out why it is being turned on and off. And what I think about in the future, where I kind of see my work proceeding, is breeders have a tremendous eye, and I think at some point we can clone it with AI. And when you have these autonomous breeding, now I'm not going to call them autonomous breeders, they're going to assist the breeders, because of course we don't want to replace the breeders. But what we want to do is basically clone the breeder's eye, and perhaps maybe have 10 to 20 of these very low-cost robots per breeder. And perhaps there's maybe some simplistic traits in early generation breeding, such as height, plant pathology traits, that if you have maybe tens of thousands of like head rows or something like that, that these robots could actually go through and help pinpoint and then track by GPS certain plants in the field. And then if the breeder isn't happy with the plants that are chosen, then he or she can just retrain the AI. So I think we're kind of at a point now when we need to be having that conversation with all the computer scientists, all of the robotic engineers on campus, to really push our science to a place that we haven't really conceived quite yet, because these things are now becoming possible, and I think Cornell is at a place where we can drive this to become a reality. And I would like to thank all of my colleagues, and with that, I will take maybe one question, so I don't want to keep you from your break, and then we can resume back at 11. All right, thank you. Yeah, Tim Setter.
We really have a lot of problems with field variability in New York. Can you say something more about how maybe routine measures or other covariates can be used in order to account for that variability and remove it as a source of variation?
When I think of my field in Aurora, New York, I want to build a robot that can go out and pick every stone out of my field. That's the first one I would do. But I think the area in virotyping is like going to be, I think, the future. So if you can think of having maybe low-cost ways to sense at the plots, like soil temperature, how much soil, water, moisture is in each plot. And then in a certain area, you can record the ambient air temperature and like other factors over the whole growing season. Then you can think about, doing factorial regression and trying to figure out what is the contribution of these changes in the E that's contributing to my G and my G by E over the whole growing season. So I think there are some things that are coming out now, but they're not cheap enough to the point where you're able just to put one probe per like one plot. So perhaps you can try to like characterize the actual variation in your field, and then use like an approach to try to like close that gap, if you will, to kind of fill in those void areas where you have no probe. So that's what I think we really need to kind of go towards that G by E question. Yeah. All right. Thank you, everyone. See you at 11.
This has been a production of Cornell University, on the web at cornell.edu.